You ever stop and think about one of life's biggest puzzles? It's something so fundamental we kind of just take it for granted, but it's the very thing that makes us who we are. We're talking about inheritance, of course. I mean, think about it. Why does an elephant always have a baby elephant? And why does a mango seed only grow into a mango plant, never anything else? And what about us? Why do siblings sometimes look almost identical and other times you barely know they're related? That right there. That's the central mystery we're going to dive into today.
So, the answer to all of this, it actually lies in the push and pull between two really powerful forces in biology. And once you get this dynamic, you've basically got the foundation for the entire science of genetics.
So on one hand, you've got this thing called heredity. That's the process that passes traits down from parent to child, and it's what keeps things consistent. But then, on the other hand, you have variation. And that's all about the differences, the stuff that makes every single one of us completely unique. And the science that tries to figure out how these two work together, well, that's genetics. You know, for literally centuries, humans knew these principles were a thing. We were already using them to breed better crops and domesticate animals. But how it all actually worked on a scientific level, total mystery. That is, until a very curious monk started asking some really smart questions in his garden.
And this is him. Meet Gregor Mendel. So back in the mid-1800s, this monk carried out a series of just brilliant experiments that literally laid the groundwork for all of modern genetics. So what was his secret sauce? What made his work so revolutionary? Well, he was the first person to bring math and stats into biology. He applied logic to a biological problem, and folks, it changed everything.
And listen, this wasn't just some weekend hobby. Mendel spent 7 years, meticulously crossbreeding and observing his pea plants. And because he used such huge sample sizes and kept incredibly detailed notes, his findings had some serious weight to them.
So you might be wondering, why pea plants? Well, turns out they were the absolute perfect subject for this kind of work. For one, they had all these super clear, contrasting traits, like you were either tall or you were short, nothing in between. Plus, they could self-pollinate, which meant he could create these true breeding lines. And maybe most importantly, Mendel could completely control who pollinated who. It was the perfect setup. And this is where Mendel's true genius really shines. Instead of trying to figure out everything out at once and getting lost in all the complexity, he simplified. He focused on just 7 specific pairs of traits, things that were totally clear-cut, either-or characteristics. And that incredible focus, it's what allowed him to see these simple, elegant patterns that absolutely everyone else had missed.
All right, so to finally crack the code, Mendel started with the simplest question you could ask. What happens if you just crossbreed for one single trait? And this, my friends, is his famous monohybrid cross.
Okay, let's break this down. First, he takes a pure, true-breeding tall plant and a pure, true-breeding dwarf plant. This is what we call the P, or parental generation. He crosses them. Now, the first generation of offspring, we call that the F1 generation. They're all tall, every single one. It's like the dwarf trait just vanished. But then, he lets those tall F1 plants pollinate themselves, and in the next generation, something really amazing happens.
The dwarf trait comes back. It reappears. And not just randomly either. He noticed a pattern. For every one dwarf plant, there were almost exactly 3 tall plants, a perfect, predictable 3-to-1 ratio. This ratio, this was the golden clue. It proved that traits weren't just blending together like paint. No, they were distinct, separate units. So Mendel came to this incredible conclusion. He figured that something was being passed down from parent to offspring completely unchanged, generation after generation. He didn't know what they were, so he just called them factors. And let me tell you, this idea, the concept of discrete little packets of inheritance, was absolutely revolutionary. Okay, so fast forward to today. We obviously have a much more modern vocabulary for the things Mendel first discovered. So let's do a quick translation of his factors into the language we use in genetics now. So what were Mendel's factors? Well, today we call them genes. Simple as that. They're the basic units of heredity, the little bits of information that code for a specific trait, like the plant's height. And for any given gene, like the one for stem height, there can be different versions, right? The instructions for tall are different from the instructions for dwarf. We call these different versions alleles. They're the reason we see all those contrasting traits that Mendel was studying. Okay, and this brings us to a really important distinction you gotta get. You have the genotype. That's the actual genetic makeup, the pair of alleles an organism has, like, say, a big K and a little t. Then you have the phenotype. That's the physical trait you can actually see, like the plant being tall. And as Mendel figured out, these two things are not always a perfect match.
And this is exactly what explains the mystery of that first F1 generation. Remember how they were all tall? It's because when a plant had both the tall allele and the dwarf allele, it still ended up tall. This means the tall allele is dominant. It completely masks the presence of the dwarf allele, which we call recessive.
And here's the truly incredible part. From these super simple experiments with just pea plants, Mendel figured out universal laws of inheritance. I mean, laws that apply to pretty much all life on Earth. These are the foundational rules of the game.
His first law is called the law of dominance, and it perfectly explains what he saw in that F1 generation. It basically says that traits are controlled by these paired units, and when you have a mixed pair, one allele, the dominant one, is going to overpower the other. So any plant with at least one big T allele is going to be tall. And this leads beautifully into his second law, the law of segregation. This is the one that explains that 3-to-1 ratio he saw in the F2 generation. The idea is that when those F1 plants made their own reproductive cells, their gametes, their big T and little t alleles separated from each other. Each gamete only got one. And because of that separation, there was a chance, a statistical probability, for two of those recessive little t alleles to meet up again in a new offspring. And just like that, the dwarf trait could pop right back up, totally unchanged.
So Mendel's work was mind-blowing for its time. He proved that heredity wasn't some weird, mysterious fluid where traits just blended together. No, it was a system of discrete, predictable units. But for all of his genius, there was one huge question he couldn't answer. What are these factors actually made of? What's their physical form? Answering that question, well, that would become one of the biggest challenges in biology for the next 100 years.